Hello, hello. Good good afternoon, good evening, good morning, wherever you are. We are Group 22 and we'll be talking about the use of synthetic biology, the use of synthetic biology techniques in tackling heavy metal pollution at Galamsey sites in Ghana. And we are a group of five people. We are a group of five people, mainly David, which is me, Sammy, Raphael, Godwin, and Bright. So as you can see from the image, Galamsey done in Ghana is done very, very poorly because we don't have the facilities and proper machinery for this kind of um, mining activity. We mostly use mercury to extract gold from the earth, and which is kind of very deadly to human beings and the environment around us. So, next slide. So, as you can see from the image, urbanization has caused a lot of, I mean, natural resources to go bad. As I said earlier, we usually use mercury to extract gold. And when the extraction of gold is done, the mercury is disposed improperly. It's just left to settle in the environment, which can cause damage to aquatic life, human life. When they go into the soil, the plants and animals absorb them, then we take them in, which can cause a lot of diseases, cancerous effects, and all kinds of environmental pollution. So we've come up with a way to kind of extract this kind of mercury from the polluted water bodies at Galamsey sites where we are. So next slide.
So I'm taking over from my friend David. So some of the heavy metals of concern, we have arsenic, zinc, copper, lead, mercury, nickel, and the likes. So these metals are heavy and they are known for their industrial and environmental presence. These metals have properties, and some of these properties are they have higher atomic weight and density. As we know, heavy metals are known for their large atomic weight and their higher densities. And also, these metals, even in small amounts, they are highly toxic, whereby even at their lowest concentrations, they're able to pose health risk to the human life and the aquatic lives. Also, looking at the environmental impact, most of these metals do not degrade or decompose naturally. Therefore, they tend to remain in the environment for long periods. And also, looking at how they accumulate in the food chain, these heavy metals build up in organisms and they can magnify as they move up in the food chain. When this happens, it leads to a greater exposure and toxicity in higher trophic levels, including humans. Going to the next slide, where we'll be talking about their impact on the ecosystem, we would look at how they affect the soil and the water. So at the mining sites, these contaminants tend to be carcinogenic, mutagenic, and they also have cytotoxic effects on organisms. Also, they also have microbial impacts where they reduces beneficial soil microorganisms and then they affect their nutrients cycling. Also, so by so doing, the presence of these toxic substances from the mining waste diminishes their populations of organisms which are beneficial into the soil. So my colleague will go on to slide 5. Thank you very much.
Sure. So we are going to talk about remediation techniques. So the remediation techniques, there are the physical, chemical, and the biological method. For the physical remediation, we have soil replacement, which is basically excavating the contaminated soil and then replacing it with clean soil from another location. And thermal desorption involves the use of heat to volatilize contaminants. Then the vapor is then collected or condensed for treatment or reuse as something else. And membrane filtration, we use semi-permeable membranes to filter out heavy metals from water, from the air, and from the soil. And ion exchange involves the use of the resin. So at the end of the extraction method or the separation, the contaminants are embedded in the resin and then they can easily be removed. For the chemical remediation, we have the precipitation where coagulants and solvents are used to mix with the contaminants to form insoluble metal precipitates, which can then be separated using simple separation techniques. Leaching involves basically washing the contaminated soil with solvent to remove the metals. Then chemical fixation involves changing the physical form or the chemical nature of the contaminants to make them less mobile or less toxic. Then immobilization involves the using of additives to bind the contaminants in place to the soil particles. This will limit their mobility and their bioavailability.
Okay, so I'll continue from where my colleague ended. So with the other remediation techniques, they end up posing toxic effects to the environment, such as the chemical. And then with the physical, it's quite expensive. So with that, biological remediation is one that comes to mind, because for most of these plants, fungi, and bacteria that are used for biological remediations, we are able to resist these heavy metals and sometimes could be found in these areas. So next slide, please. Next slide. Okay, so I'll continue. So with our work, we are focusing on mercury, and for mercury, there are mercury-resistant bacteria such as the Bacillus subtilis, the E. coli, Pseudomonas, and several other Saccharomyces cerevisiae, which are all microorganisms that have been found to be able to eliminate mercury from their environment because of their metabolism that is able to change usually the mercury from its toxic form to the non-toxic and atmospheric form. Yes. So basically there's a picture with a redox reaction where mercury is being reduced, the toxic reaction. The toxic form is being reduced to a non-toxic form through redox reaction, and others also through carrier molecules like the NADP and FAD interaction with the cysteine. Yes. So all these are mechanisms that exist for mercury remediations. Yes. And to continue with it, so synthetic biology approach, what happens is that we could look at this Bacillus subtilis, E. coli, and Pseudomonas, which we will be able to engineer them to be able to carry these MER genes. The MER genes, specifically the prominent ones that have been known to do the remediation, being MER-A and MER-B, are very important to produce the mercuric reductase enzymes and the organourea lysis, which have been known to help remediate mercury in their environment. Yes, so these are expression systems that can be used and can be leveraged on for the synthetic biology approach of it. And we could look at how, because they already also exist, they are already existing strategies. Can you move on to, so thank you, can move on to the next person.
Oh, okay. I'm also Bright, and I'll also be taking you through the biosafety consideration. So basically, with the biosafety technique, it tends to prevent the risk of exposure to these pathogens or these microorganisms, and also helps to mitigate them from leaking out into our external environment through the use of the biosafety cabinet, the HEPA filters, the airlock system, and the use of PPEs. So with a containment strategy, we tend to incorporate into the organism the genetic circuits and the kill switch, which are all stable when the organism is in a controlled environment. So as soon as the organism found or tend to leak into an uncontrolled environment, the 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 circuit activates and turns on the kill switch to help to deactivate these microorganisms. That might cause serious harm to the public and also to the ecosystem. Moreover, with the conclusions and future prospects, with the scaling up process, so per the paper that we read, as my colleague earlier said, we found out that the MER A and the MER B gene were able to detoxify the mercury to a less toxic form. So in doing this, you have to scale up these genes. We have to produce mass number of these microorganisms so that we can deploy them to these various sites to bioremediate these heavy metals from our environment. And we found that one of the challenges that we are mostly encountered are the compliance to biosafety regulation, the high cost of advanced bio-containment facilities, and etc. And also the strategies are opposite of the challenges. We have to comply by the biosafety regulations and so on. So with the future prospect, we tend to we are going to combine the conventional technique with the biological technique to help in bioremediating these heavy metals faster from our environment. So with the use of only one technique, the bioremediation seems to be very slow. So with the combination of the two technique, it will be able to bioremediate these heavy metals from our environment and prevent it from causing serious harm to the ecosystem. Thank you. So you are group 22 and represented on this.